Hello, my name is Colin and I'm here to guide you through a pretty sweet lesson. After all, it's about cyclic carbohydrates. We'll start by reviewing some relevant organic chemistry, like the hemiacetal formation reaction. We'll use that to illustrate how sugars form rings. We'll examine the conformation of those rings. And lastly, we'll investigate anomers and the process known as mutarotation, all so that we can score more points on test day. Let's begin. First, we always want to start with a learning goal. And our learning goal with this review is to get to a point where if we see a cyclic carbohydrate on test day, we can look at it and say, I know what that is, and I know how that was formed. And we always want to be thinking about questions. We want to be able to answer questions like this one that give us a mutarotation reaction and ask us what the probable intermediate is for that reaction. Before we can get to this point, we need to review the relevant organic chemistry. That is the hemiacetal formation reaction and the Hemiketal formation reaction. If we understand these two reactions, we'll be able to understand how sugars cyclize on test day. Let's look at the hemiacetal formation reaction in action. To form a hemiacetal, we start with an aldehyde on the left and an alcohol on the right. The alcohol functional group of the alcohol will attack the carbonyl carbon of the aldehyde, forming a species with charge. We should know for test day that molecules with charge are high energy, they're unstable, and they'll get rid of that charge if they can. That leads us to the second step in our hemiacetal formation mechanism. Our intermediate will get rid of its charge, forming the hemiacetal. The hemiacetal has 4 different functional groups on its central carbon: an alkyl group, a hydroxide group, an alkoxide group, and a hydrogen. You may note that we have a carbon with 4 groups and therefore a chiral carbon. We form a chiral carbon when we form a hemiacetal. The same is true when we form a hemiketal. The hemiketal mechanism is much the same as the hemiacetal mechanism. The difference is you start with a ketone rather than an aldehyde. Otherwise, the reaction proceeds almost the exact same way. Knowing this can save you some time when preparing for test day. While these reactions are certainly great to know for the MCAT in general, we wouldn't be talking about them here unless they applied to sugars, and they do. We'll illustrate that with D-mannose. Notice that D-mannose has a hydroxide group in green and an aldehyde group in orange, both of the components you would need to form a hemiacetal. When D-mannose cyclizes, that's exactly what it does. It internally forms a hemiacetal. Notice that at the top we have a carbon with an alkyl group, a hydroxide group, an alkoxide group, and a hydrogen, just like the carbon we formed in our hemiacetal formation reaction. That's because the cyclization of D-mannose is a hemiacetal formation reaction. This D-mannose started with one aldehyde group, but it had several different hydroxide groups. Depending on which hydroxide group had attacked the aldehyde group, it could have formed different rings. Not only should we educate ourselves about these rings, but come test day, we want to be able to classify them as well. Pyranoses would be a good place to start. When sugars cyclize and form a 6-membered ring, we call that a pyranose. We should know for test day that 6-membered rings are relatively stable and therefore fairly common in nature. Thus, the pyranose is a common form of a cyclic carbohydrate. Here we have 3 different depictions of a 6-membered ring formed from the cyclization of the sugar galactose, which would therefore be called a galactopyranose. On the left, we have a Fischer projection with its 6 different carbons labeled. In the middle, we can see those same 6 carbons on the Haworth projection, as well as those same 6 carbons on the right when this ring is in the chair conformation. We should know as background that 6-membered rings prefer to be in the chair conformation because it's the lowest energy. And since pyranoses are 6-membered rings, they too prefer to be in the chair conformation. Equally important are the furanoses. The word furanose is used to describe 5-membered rings, such as the D-fructofuranoses that we've depicted here. On the left, again, we have a Fischer projection of our fructofuranose with the 6 carbons labeled. Notice that it has 6 carbons, but it's only forming a 5-membered ring. Be aware that just because you have 6 carbons does not mean you necessarily must form a 6-membered ring. On the right, we see those same 6 carbons labeled on the Haworth projection. We don't see a chair conformation here, though, because 5-membered rings don't form the chair conformation. Not only can we form different size rings, but every time we form a given ring, we can form it in 2 different ways, leading to even more variability, which brings us to anomerization. In order to investigate anomerization, we've depicted a Fischer projection of a glucose molecule. When this glucose molecule cyclizes, a hydroxide will be attacking the carbonyl carbon of an aldehyde. Well, an aldehyde is a planar functional group, and the hydroxide could attack it from either side. Because of this, we could form two different types of cyclic glucose molecules. On the top, we have an alpha D-glucose. The C1 carbon has a hydroxide group pointing down, whereas on the bottom we have a beta D-glucose, where the C1 carbon has a hydroxide pointing up. That makes these two molecules epimers, but they're a special type of epimer. We call them anomers, and that carbon is known as the anomeric carbon. We want to be able to look at this molecule in different ways, so in addition to having the Haworth projection, we'll also draw the Fisher projection on the left and the chair conformation on the right. If you draw a cyclic D-glucose in this way, with the C6 carbon pointing upward, something nice happens. In the alpha form, the hydroxide group is pointing down. You can think alpha looks kind of like a fish, so this hydroxide group is pointing downward like a fish trying to get into some water. On the other hand, the Greek letter beta can remind you of a butterfly. The beta D-glucose will have a hydroxide group pointing upward like a butterfly trying to get into the air. It's always nice to have tricks to remember things. It turns out cyclic sugars are fickle molecules. If you have a pure crystal of alpha D-glucose and you throw it in a solution of water, pretty soon you'll have a mixture of alpha D-glucose and beta D-glucose. That's because alpha D-glucose can uncyclize, forming the straight chain glucose shown here in the middle. If that straight chain glucose recyclizes, it could either proceed to the left and reform an alpha D-glucose, or it could proceed to the right and form a beta D-glucose. The result being that as a population of glucose molecules in a solution cyclizes and uncyclizes, some is bound to be in the alpha form, while others are bound to be in the beta form. This is because the anomeric carbon only has chirality when the sugar is in the cyclic form. When the sugar uncyclizes into the straight chain form, that carbon simply becomes the achiral carbon of a carbonyl. In addition, it has no memory of what it was before. The next time the sugar cyclizes, that carbonyl carbon could be struck by either side. After all, it's planar. Make sure to know the term mutarotation for test day and why it occurs. Now let's practice some questions. Go ahead and attempt question number 1. If you chose answer choice D for question number 1, give yourself a pat on the back. Ribose, as we can see on the right, is an aldose sugar. It has an aldehyde functional group, not a ketone functional group. Aldoses should form hemiacetals when they cyclize, not hemiketals, as in answer choice D. Therefore, answer choice D is false, which makes it our correct answer. Next, attempt question number 2. We should be able to identify this reaction as a mutarotation reaction. These two molecules are the same, except for their anomeric carbons. Mutarotations change the anomeric carbon. We can see that we have one anomer on the left of the reaction arrow and a different anomer on the right. We just need to find the correct intermediate carbonyl, which is on answer choice A, the aldehyde. Answer choice B had a carboxylic acid, answer choice C had a ketone, and D had a diol, none of which were the correct intermediate for this question. Takeaways. Sugar cyclizations occur through hemiacetal or hemiketal reaction mechanisms. This results in the formation of a new chiral center, and therefore, whenever sugars cyclize, you can form alpha or beta forms. The anomeric carbon is derived from the carbonyl carbon and can be seen in every form of the molecule, while cyclic sugars can exist in the alpha or the beta forms. Neither one necessarily exists forever. Alpha and beta anomers can interconvert through the process known as mutarotation. Short and sweet, that concludes our review of cyclic carbohydrates. Keep up the hard work, it's going to pay off on test day.